Fine. So we saw about the enzymes that are required for replication in the last class. Now we'll see about how this DNA is going to make one more DNA, and we'll see the steps one by one. Is it clear? The first step that is involved in DNA replication is activation of nucleotide. So quickly, in a very simple way, I'll tell you what is a nucleotide and how a nucleotide is forming a nucleic acid. Very quickly, I will tell you. Nucleotide means a nucleoside plus one phosphate. A nucleoside plus one phosphate is called a nucleotide. So many nucleotides together. One nucleotide, along with that another nucleotide, like this. If I keep on adding, it becomes an oligonucleotide. Many nucleotides, polymer of nucleotides, or you can call it a chain of nucleotides. If it happens in that way, then you call that oligonucleotide. This oligonucleotide is a nucleic acid. This oligonucleotide is a nucleic acid. It can be a small nucleotide, it can be a large number of nucleotides, but that functional nucleotide is called as a nucleic acid. Okay. The first step in DNA replication is activation of nucleotides. Activation of nucleotides. So what is this activation? We'll be seeing it in further processes. But for now, you please remember this thing. We have got the 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. We have got these 4. Here uracil will not come. Why it will not come? Because that is only in RNA. But since we are talking about DNA and DNA replication, one DNA is producing another DNA. So there is no job for uracil here. So uracil will not come here. So adenine, thymine, cytosine, and guanine. All these 4. Adenine monophosphate, or it is called as adenosine monophosphate. Thymine monophosphate. Then it is cytosine monophosphate. Guanine monophosphate. All these monophosphates, they are all called as monophosphates because they will have only one phosphate with them. They will get converted, they will get activated to adenosine triphosphate, thymine triphosphate, cytosine triphosphate, and guanine triphosphate. Now they will get 2 or 3 phosphate molecules get added to that AMP and TMP. Now what is the exact chemical pathway that is happening in between? That we'll be seeing later. When we are seeing the further sequences and further processes, we'll be seeing that. But for now, you just remember the first step in DNA replication is activation of nucleotides. Each and every nucleotide will get activated, and the enzyme that facilitates this activation is phosphorylase. And you all know what a phosphorylase does. It adds phosphate. Phosphorylase actually keeps on adding phosphate. So number of phosphate it keeps on adding. The number of it becomes diphosphate, triphosphate, decaphosphate, pentaphosphate. It keeps on increasing. Okay, children. So the first thing is activation of nucleotides. And for all the 4 processes, phosphorylase is only the common enzyme. Is it clear? The second step after the activation of nucleotides is opening the DNA helix. You all know how a DNA will be. This is how a DNA will be. This DNA has to be first unwinded. This is a helical model, helical structure. It has to be unwinded. Unwinded means what? Am I right? This has to happen. Who does this? The enzyme called helicase does this. The helicase wants to come and act. Helicase will come and wait. Now, where will the enzyme act? Which place the enzyme will act? Helicase has to act somewhere. Where will it act? That depends on ORI site. Origin of replication site. This which we have already studied. This is a particular sequence from where the replication of the DNA starts. It is a sequence. It can be AT, GC, It depends on different kinds of DNA. So a particular kind of DNA will have a particular ORI site, the origin of replication site. The helicase will go and start removing the helical structure only at the origin of replication site. Is it clear, children? So this ORI site, if you take in case of a prokaryote, there is only one ORI site. But if you take eukaryote, there are more than one ORI site. Is it clear? So helicase will go there, and what will happen? The helicase will act at that particular place, and it will open the helix, which means this helical structure is broken. How does it do? It breaks the hydrogen bonds in between. We saw that in enzymes. Do you remember? It breaks the hydrogen bonds in between, and you get 2 linear strands of DNA separately. Then what happens? There will be some tension and stress in the strand. Topoisomerase comes, it cuts, it rotates and pulls off the strand separately, and all the tension goes off. And after that, they have to make further sure that single strand stabilizing protein or enzyme comes act individually on the strand to make it completely free of tension. Now we have two strands separately. So this is done by the enzyme. Now this is very important. Listen here, children. Action of A. First, recognition of ORI site. Helicase enzyme is activated and it has got acted on the DNA, and finally topoisomerase. All these three are finished acting. So action of A, B and C on DNA makes the DNA into a model called as replicating fork. It makes it into a model called as replicating fork. Now let us see what is happening. This is the double stranded, okay? This is the double stranded DNA which has been separated by the helicase enzyme. When I keep it here, helicase enzyme is coming here. Helicase enzyme is coming and it is going on acting. When I send the helicase enzyme like this, what happens? The DNA strand completely keeps on separating. And at the back of it, when the helicase is acting itself, at the back, topoisomerase is acting. So the strands are completely even more separated. And when the topoisomerase is acting, at the back itself, the single strand stabilizing protein is also coming. So all the three are parallelly going, one behind the other. So what happens? The DNA keeps on opening up like this. Am I right? It keeps on opening up like this. So we get this kind of a model. Is it clear, children? Now, what I am going to do further is 3' and 5'. 5' and 3'. Suppose this is the polarity of our DNA strands. I have just drawn the same thing here. 3' and 5', 5' and 3'. Okay. Now, the main enzyme that is required for DNA replication is DNA polymerase. Now, this DNA polymerase will attach in this position. Only from this position, new strands of DNA are being synthesized. So, when the new strands of DNA are being synthesized, they will be having another polarity. They will not be having the same polarity as this end. Am I right? So, just for what do you call a recognition, for our own evaluation, our own recognition, I am naming it as 5' and 3', because it will surely be not of 3' here, because new strands are being produced. So I am just naming it as 5' and 3'. Is it clear till this? Shall I go a little further? Very good. Now what happens? After this step is over, after the replicating fork model has come, the third step comes. Primer formation. The primer is being formed. Now we'll spend some time understanding what is a primer, how it is bonding, and what happens in the formation of core replication. Is it clear, children? The first thing you need to know about primer is a oligonucleotide. In the starting of the session, I told you, do you remember? It is a oligonucleotide. Oligonucleotide means one nucleotide, second nucleotide, like this large number of nucleotides added, one behind the other is a oligonucleotide. It is a oligonucleotide of a RNA. Primer is a oligonucleotide of a RNA. Is it clear, children? And one end will be a 5' and one end will be a 3', just as the DNA. You find here 5' on one end, 3' on one end, 3' on one end, 5' on one end. What do these actually represent? 3' actually represents OH, a free functional group, and 5' represents a phosphate group. That is what I have written here, 5' and 3', and 5' represents a phosphate and 3' represents a OH group, a functional group. Is it clear, children? So this primer is first being formed. I already told you this primer formation is done with the help of an enzyme called as primase enzyme, and this primase enzyme is a DNA directed RNA polymerase, which means the primer which has to be produced, what kind of primer has to be produced is decided by the DNA which is undergoing replication. If this DNA is undergoing replication, what primer has to be produced for this DNA replication is also decided by the DNA itself. Is it clear till this, children? Okay, now primer is being produced. After the primer gets produced, this is the structure of a primer. Okay, I can put it like this, but for our convenience, we are putting it as one big rectangular box. Okay, one end is 5', it consists of a phosphate, Other end is 3 dash, it consists of a OH. Now this primer will come to both the strands of DNA, children. It will come to both the strands of DNA. And you should always know, we saw this in the nucleotide formation also. The bond is always formed between the 3 dash and the 5 dash. That is the phosphate and the OH. Bond is always formed between phosphate and OH, and the name of this bond is phosphodiester bond. Am I right? The same kind of bond pairing happens here also. What happens is, the primer comes and sits in the place that 3 dash is. Because here it can form bond only between 5 dash and 3 dash. Am I right? So it comes and sits here. There is a bond that is formed between 5 dash and 3 dash at this position. Okay? The primer comes and attaches there in this strand. But in the opposite strand, it attaches down. Because only here 3 dash is there. So one more phosphodiester bond is formed at this position. Is it clear? So two primers have come and attached now. Primer has been formed and primer has come and attached. Is it clear till this? Now what happens? There are nucleotides freely suspended in the cytoplasm. Nucleotides. There are nucleotides suspended here and there. Now both of these primer will bring one nucleotide and attach to the primer and so on. Now let us see what is happening. The first nucleotide is coming. The first DNA nucleotide is coming. When the first DNA nucleotide is coming, the DNA nucleotide will also have the same structure. It will be like this. It will have 5 dash on one end, 3 dash on one end. Now the primer is having a 3 dash here. Am I right? So if a DNA nucleotide comes and attaches to this 3 dash, it means there will be a bond that will be formed between 5 dash and 3 dash, and that will be a phosphodiester bond. Is it clear? So again, this end is 3 here. This 5 dash and 3 dash is joined, and this end is 3 here. Again, one more nucleotide will come. That will again form a bond here, phosphodiester bond. And again, one more nucleotide will come. That will also form one more phosphodiester bond here. So what happens is the nucleotide keeps on adding. The DNA nucleotide keeps on adding at this fragment. Now let us see what happens in this fragment. In this fragment also the same thing happens. Because 5 dash 3 dash is here. One DNA nucleotide will come. That will have 5 dash 3 dash. A phosphodiester bond will take place here. Another phosphodiester bond will take place here. So it keeps on adding. So one nucleotide behind one nucleotide, it keeps on adding. Is it clear? Now, although it is keeping on adding to that end, how is it attached to this end? What is the bond that is taking place here? It is attached by normal hydrogen bonds. Normal hydrogen bonds, it is attached in these areas. Is it clear, children? So, because of this phosphodiester bond, one nucleotide behind another nucleotide keeps on getting added, and this series of nucleotides gets attached to the parental strand. This is the parental strand, right? The main strand by means of hydrogen bond. Is it clear, children? Now, let us go a step further. Okay, now what happens? Helicase is acting further. Okay, please listen. Helicase is acting further. Topoisomerase is acting further. Single strand stabilizing enzyme is acting further, which means it is keeping on moving. It is keeping on moving inside. So the fork becomes enlarged. The fork becomes enlarged. When the fork becomes enlarged, I am not doing anything. Earlier, 2 were there. The same 2 I have drawn here. Again, one more nucleotide will come. That will be 5 dash and 3 dash. One more nucleotide will come. 5 dash and 3 dash. Again, a phosphodiester bond will be formed here. Phosphodiester bond will be formed here. Hydrogen bond, hydrogen bond, hydrogen bond, hydrogen bond. This side, no problem. The oligonucleotides keep forming upon this side, the DNA oligonucleotides. So now you come to this side. 2 oligonucleotides are formed. I have drawn the 2 here. Okay. Now the primer is here. The first nucleotide has come, second nucleotide has come. Now, is there a possibility of another nucleotide to come in that position? There is no possibility to come because there is no parental strand there. The parental strand got over here. The parental strand has got over there. Am I right? So what happens? One more primer comes and attaches at this end. One more primer comes and gets attached at this end. And again the same process starts. 3 dash, 1 5 dash comes here, 3 dash, 5 dash, 3 dash, again phosphodiester bond, hydrogen bond. Again it keeps on adding. So what happens is, in one strand there is a continuous synthesis, but in the other strand there is a discontinuous synthesis. Is it clear? In one strand, if you see, there is no breakage at all, because the parental strand is going from 3' to 5'. So it will have no problem. But when it is going from 3' to 5', it will start at one particular position, it will stop here. Again, it will start from this position, it will stop here. And after some time, when the helicase goes further, when the fork becomes further large, again this will come a little bit down. Is it clear, children? So what happens is, in one strand there is always a continuous synthesis, and on the other strand there is a discontinuous synthesis of the nucleotide. The strand which has continuous synthesis of nucleotides is called as the leading strand. It is called as the leading strand. It will lead the way. The one which does not have this continuous synthesis, which has got discontinuous synthesis, it is called as lagging strand. Is it clear? It is called as lagging strand. Now, one more thing you should understand is the new strand that is formed. Now, this is the DNA nucleotide that is formed. Am I right? This is the DNA nucleotide, but we have not made it a strand. We are yet to make it a strand. How the two strands are coming, I didn't tell about that. I'll be telling you. This new nucleotide is always formed in 5' to 3'. This is very, very important. The new strands in DNA are always formed from 5' to 3'. You can find 5' here. It is going towards 3'. In the same way, it is 5' here. It is moving towards 3'. It is always formed from 5' to 3'. And the parental strand is always from 3' to 5'. The reason is, the primer always comes and attaches only through the 5'. The primer always gets attached only to the 5', because the phosphodiester bond forms only between phosphate and the OH. Is it clear, children? So the new strand is always synthesized from 5' to 3', and there is one leading strand and one lagging strand. Leading strand is a continuous synthesis and lagging strand is a discontinuous synthesis. Is it clear? Now, whatever replication fork model that I told now is applicable only for linear DNA. Linear DNA. If it is a circular genetic material, circular DNA, prokaryotes, for example, then that is not, this process is not at all applicable. It is completely separate. They will not form a replicating fork. Instead, the origin of replication, the helicase enzyme will act and it will form a bulged structure like this. It will form a bulge structure, and that is called as replication bubble. So you should always keep this in your mind. Replication fork is possible only in linear DNA. In prokaryotes, in circular DNA, generally, this is not at all possible. Okay? So, we have got the leading strand and lagging strand, but we are yet to separate it completely. So, we will see what is happening when they get separated and how these fragments are being joined together and what are these fragments further. Okay? Copy it.